snaps, snaps, snaps. Jazz hands.
It's a snap clap. A snap clap.
That's it. I like the snap clap. Also known as a slap.
Welcome to Absolute Genius, a podcast series from Thermo Fisher Scientific. I'm Jordan Ruggieri.
And I'm Cassie McCreary, and it's our pleasure to welcome Dr. Leanne Baxter to the show as our esteemed genius of the day.
Leanne is the director of the HLA Laboratory at Children's Hospital Los Angeles, and she has contributed to an incredible amount of research and scientific discovery over her decades-long career. We're so thankful to learn from her about both her research and her clinical work, as well as the incredible stories about how her innovation has impacted lives around her. Thanks so much for joining us today, and we hope you enjoy. Leanne, it is a pleasure to have you with us on Absolute Genius today. We're super excited to have you on. Your research is really, really interesting. Can you maybe just give a little bit of background on yourself and what type of research you're involved in?
For the most part, I work in transplantation. And I came to this field many decades ago because of my interest in being able to develop new technologies and new laboratory tests that could be used ultimately in clinical care, although my work is at the research stage. Eventually, actually, I evolved into having dual roles. I have a research laboratory, and I also have a separate clinical laboratory. And one of the advantages of that is that it allows me to translate some of the things I do ultimately into practice.
That's amazing. Can you elaborate? How does this research work? Are you looking at how a subject might accept or reject a transplant? Can you elaborate on that a little bit?
Most of my work involves compatibility in some way. In the early days of my work, I did research to better understand matching of HLA between donors and recipients and how that impacts blood and marrow transplants. And that over decades has evolved into standard practice for testing that's done now by sequencing, nucleotide sequencing. But I was in the early days of the work, even involved in sequencing some of the alleles and developing methods. So it was just a great fun for me and very rewarding. And then to see it ultimately move into clinical practice and change lives has been extraordinary. I feel very lucky. Today, I'm still working with compatibility. Some of the work that we've recently published involves understanding why the HLA alleles in the population differ in ethnic groups and how that impacts the ability of a clinical laboratory to measure certain endpoints in clinical tests that are important for assessing compatibility. But I'm most excited about a project we're doing to look at cell-free DNA after transplantation. Cell-free DNA in itself is absolutely amazing and provides a lot of opportunities for future diagnostic tests. There are some diagnostic tests already available for cell-free DNA, but I believe that we've only just touched the surface of what's possible there.
Cell-free DNA is amazing. What is cell-free DNA, and what exactly do you look for, in that particular type of DNA?
Cell-free DNA is something that gets released into the circulation when cells die. And the nature of that cell-free DNA can vary depending upon the process that caused the death of the cell. So that's one of the opportunities to get additional information. In my laboratory, we're looking for assays that can allow us to distinguish donor-derived cell-free DNA from host-derived cell-free DNA. There's a lot of interest in using donor-derived cell-free DNA and the levels of donor-derived cell-free DNA to detect when an organ that's been transplanted is being damaged. And there's growing evidence that there is a correlation and that perhaps, monitoring the levels of donor-derived cell-free DNA could be useful to detect organ damage very early in the process. That would allow early intervention to prevent the severe damage from ever occurring, and we think ultimately can result in much longer graft and patient survival.
Is there any challenges looking at donor-derived cell-free DNA versus host cell-free DNA?
Tremendous challenges. That's what makes it fun. It's extremely challenging. First, the levels of cell-free DNA in the blood are very low, 0 to 100 nanograms per mil, very little. In a situation such as ours, we're interested in studying pediatric samples, so we want to be able to get results from a milliliter of blood. So that means we have to have an assay that can work with very little DNA input. Beyond that, when you're looking at the amount of donor-derived cell-free DNA in the midst of all the host cell-free DNA, there isn't very much most of the time. So if you have a healthy graft, some of the literature suggests it might be as low as 0.2%. So you can imagine why I think digital PCR is a godsend.
Yeah, definitely. How is this looked at, let's say, prior to digital PCR? How, and even maybe some of these techniques, how would somebody determine that there might be a response against that transplanted organ? Is it done molecularly, or is it done maybe proteomics, immunology? How does that work?
Historically, well, actually still, a standard practice would be to use some sort of markers or indicators of organ function, and they're very insensitive. So in the case of kidney transplantation, serum creatinine is used as a biomarker, and then an invasive biopsy is generally used to determine if there's some pathology in the organ. There's a lot of new things coming out. There are assays that are looking at RNA either in biopsy material or in blood, and looking at whether or not they can be useful biomarkers. It's a rapidly advancing field and very exciting. It's providing new insights into the diagnosis of rejection, the pathways involved in rejection, and ultimately, we believe that it's going to lead to better therapeutic interventions, as well as preventing damage in the first place.
I can just imagine. Talking a little bit more about some of your workflow, can you talk a little bit about how the assays work? Are you looking at a particular biomarker for, say, donor-derived, then you are looking at host-derived? Is there any multiplexing or somewhere around there?
That's exactly what we've been working on, is multiplexing and developing primers and probes that can be used to differentiate donor and recipient. We are using indels, insertions and deletions, and the alleles that we're using now have been selected so that they're between 0.5 or 0.6 frequency in the population. The goal there is to have as many informative markers as we can. And we started by trying to develop a whole new approach to digital PCR that allows us to multiplex beyond what's been available before. We've recently published a paper in Frontiers in Genetics describing this approach in detail. Interestingly, what we did in the first demonstration is that we could detect 12 different targets in one well instead of one. It would be very difficult to do, but we think it could be done, that you could get up to 25 targets per well. And then, if you use, multiple wells for one sample, then you can also increase the number of targets beyond that. But in measuring donor-derived DNA, it's my observation that having many targets, and being able to average them is beneficial because you're working at something that's just such a low level, and it improves precision and accuracy when you have multiple informative targets.
Yeah, makes sense. Let's transition over to digital PCR. We love digital PCR at Absolute Genius. I'm sure you know. How does digital PCR play a role in your research?
For us, we think it's perhaps the only useful platform to be able to test samples that have only 1 milliliter of blood and to isolate cell-free DNA from them. We're not going to have very much DNA to work with, and we're going to be measuring very small quantities. And when you have a very low copy number of something in a mixture, digital PCR is really the best approach. It helps you to get a much lower baseline. For some of the probes we've been working with, if we just see one positive signal, we can be pretty confident that there's one copy there.
You mentioned NGS and using some NGS as kind of an industry standard or a direction that the industry is going in. Is there any differences or do they any differences between the two in terms of your research? Or are they complementary? How could you maybe use NGS versus digital PCR? When might it be useful to use one or the other?
Clinically, we're using NGS. Our research involves the digital PCR. So, our long-term goal would be to see this technology move into the clinic.
And is it simply the ease of use and that digital PCR offers or turnaround time? Like what are some of the benefits of using digital PCR?
The list is long. The ease of use is amazing. For our research assay, the setup is 30 minutes, and it doesn't require much expertise at all. One of the students working in our lab had never taken a lab course. She was a major in public health, and she wanted to volunteer to work in our lab. We were very fortunate to have her, and her first assay looked great.
Amazing.
Yeah, so it's really easy to use. Another thing is the speed. Here, we can just do one sample at a time if we wish. And for the assay we're working with, it takes about, if we had a whole plate of 16 samples, it would take 30 to 60 minutes to set up and 2 to 2.5 hours to run. But if we have only one sample, it's 30 minutes and a shorter run time as well.
That makes sense. I can imagine too, holding samples, especially cell-free DNA, you run into issues with potential freeze thaw and degradation. And is that something you come up against frequently as well?
We've been, I think, successful at being able to store our samples, but we're looking for something that ultimately will be used clinically. And one of the drawbacks of a lot of the current options are that it takes days or longer to be able to get the results. So when I've discussed our research at clinical meetings, I can't tell you how much positive feedback I've gotten from the clinicians saying, having a true same-day assay would be extraordinary, and it would really change their ability to manage patients.
Can you talk a little bit about multiplexing as well? How does that look on digital PCR as opposed to maybe a real-time PCR platform or a different platform?
The work we're doing is in collaboration with a company who holds the patent for this technology, and we've been the first group to be able to explore it. I'm really thrilled about having that opportunity. It's an amazing probe design that allows us to multiplex. And the way it works is that the probe itself forms a hairpin, and the target region of the probe has a ribobase incorporated into it. At the 5' end, there's the dye that we use to detect the presence of the probe. And when there's no target present, it's in this partial hairpin conformation, and you detect the fluorescence. Then when you go through cycling, what you can do is denature the hairpin loop in the presence of your target DNA. When it hybridizes to the target, that presence of the ribobase allows cleavage by ribonuclease. Then after the cleavage, that hairpin loop piece comes off and the other part stays with the target DNA. Now you add polymerase and it extends it. And that dye at the 5' end has an iso-C. And in this polymerization, you add an iso-G with a quencher attached. So now you're going from a signal to no signal. Quite different than most of the technologies that are out there. But because we can control the melt temperature, we can use several different probes that melt at different temperatures during a single cycle.
Very, very interesting. Anything else science-wise or digital PCR-wise that we might have missed or that you wanted to talk about?
No, I really like working with technology, and I feel very fortunate that for most of my career I got to do this. This is my idea of fun. And the wonderful aspect of the area that I work in is that a lot of my work has ultimately evolved into clinical practice. And, knowing that it's affected the lives of people is really rewarding.
We hope you're enjoying this episode of Absolute Genius. We are huge fans of digital PCR here and want to tell you about Applied Biosystems' QuantStudio Absolute Q dPCR system. This instrument enables quantification of your targets without the need for standard curves with excellent precision in only 90 minutes.
You can say that again, Cassie.
Okay, I will. Get dPCR data in only 90 minutes.
Unlike other digital PCR systems, the Absolute Q dPCR instrument does not use emulsion or other droplet-based methods to compartmentalize reactions. Rather, using distribution channels on a microfluidic array plate, the reagent is consistently delivered to physical microchambers. The technology utilizes more than 20,000 microchambers with a coefficient of variation of less than 1%.
Another feature we're excited about is that the Absolute Q dPCR system analyzes more than 95% of the loaded sample, ensuring highly accurate digital PCR results. No more wasting upwards of 60% of your precious samples when loading, which is especially critical for rare targets.
Yes, and Thermo Fisher Scientific has developed a portfolio of robust Absolute Q digital PCR assays. So if you need precise quantification of gene targets for research applications like AAV viral titer for cell and gene therapy research, eDNA analysis, SARS-CoV-2 wastewater surveillance, or liquid biopsy mutation and monitoring, then you'll want to look into this for sure. You can learn more at www.thermofisher.com/AbsoluteQ or visit the Absolute Genius webpage. Again, that's www.thermofisher.com/AbsoluteQ or visit the Absolute Genius webpage. The Applied Biosystems QuantStudio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures.
Now, back to our episode. Woo! Leanne, welcome to Cassie's Career Corner. We just talk about fun, warm and fuzzy career things and advice and all kinds of fun stories. So, we've spoken a lot about your passion around this and how much it excites you and how happy it makes you and everything. What is it about it that you're so passionate about, or what is it that you so connect with? And was there anything early in your life that made you think kind of more broadly, I really want to go down the route of science and research?
When I was a little girl, I didn't want to play with dolls. I loved experimentation, not like having a kit, but actually observing the world around me and categorizing it. I used to do things, my favorite thing to do was to go to my aunt's farm, and she had a lot of cats. So my cousin and I would collect all the cats and then test them to see what would they eat. And then make records of it, or what was their tolerance to various noises or various stimuli.
I have one right here. I'm going to do that later.
And that was my idea of fun. I think there's something to be said for we're each meant to do something. And maybe you can even go back to your early parts of your life and think about what was a fit and have that guide you into a career. For me, I also like writing a lot. And then when I went to school, I was actually an editor on my college newspaper as well. So I was thinking about a writing career, but then I love science. And ultimately, the more involved I became in science, there really wasn't time for the journalism part of my inclination anymore. When I started as a graduate student, I was really interested in enzymatic pathways and their regulation and hardcore biochemistry. But I loved everything I learned about, with two exceptions. I thought antibodies were pretty boring. And I also wasn't very interested in nucleic acids either. So those were like the only two areas I thought I could say never. And then I actually became interested in gene regulation, understanding, some enzymology that was created, some changes that were related to alternative splicing of the enzyme itself. And that led to really becoming immersed in molecular biology and learning a lot of technology. And all of a sudden, that became my passion. So much for not being interested in molecular biology. That one was gone. So I was doing molecular biology. That's what brought me to this field of HLA. At the time that I entered the field, in transplantation, differences in HLA between individuals were determined using serological methods. And I entered the field at just the right time because the molecular biology hadn't yet been explored to any great extent. And so I had the opportunity to be involved in developing molecular methods for doing typing that had historically been done by these serological methods. That led to incredible discoveries, not by me personally, but this large field of people pursuing the same technology. What we thought used to be maybe 100 types turns out to be millions of alleles. And all of this is important in clinical transplant settings and even other settings like a person's ability to fight an infection. It's really been a lot of fun. But in working in transplantation, another part of compatibility is that the immune system can develop antibodies against the graft. And as my clinical lab was evolving, we were also testing for antibodies. So I also became passionate about antibodies. And so now, if you look at my publication record over the last few decades, it's all either molecular biology or antibodies.
Isn't that funny how that works out? So going back to the beginning a little bit, what did you study in school? Because you said kind of at the beginning you were more heavily steeped in the biochemistry side of things. Is that what you studied, or?
As a graduate student, I worked in the lab of Hector DeLuca, who was responsible for discovering some of the vitamin D metabolites.
Oh, awesome.
And we were doing some of that work while I was a graduate student. And so I was doing assays, studying the hydroxylases for vitamin D. There's a 1 hydroxylase, a 24 hydroxylase, and a 25 hydroxylase. And so I was studying those, and it was a really exciting time. When I finished my graduate work, I started thinking that I planned to stay in the vitamin D field, but I thought that maybe vitamin D was like estrogen in that it regulated gene expression. So then I did a postdoc in a lab looking at regulation of gene expression by estrogen, which is what brought me to molecular biology.
There we go. It's all connecting. So kind of a theme throughout our show has been the discussion of the importance of finding mentors over your career and your studies, or networking or things of that nature. Would you say that any of that has played a crucial role for you over the course of your path?
Oh, certainly. I think my advisors through my training definitely played a role. And then later, especially when I took a deep jump into this HLA field, I was actually surprised to get hired to work in this field. What happened is that I had been doing a lot of molecular biology, and I had moved away from the regulation and things that were potentially clinically relevant to some more hardcore enzymology and looking at these isoforms. Although I was doing well, I didn't feel as passionate about what I was doing as some of my earlier work, and I decided I wanted to do something that had more clinical relevance. And so I started thinking about my skill set and looked at this HLA situation with the typing, and I thought there was this fabulous opportunity to apply molecular biology to HLA typing and also to understanding what the difference was between those HLA types. I applied for a job at the Blood Center of Southeastern Wisconsin to establish the first clinical laboratory to do DNA-based HLA typing. And they hired me, even though I knew nothing about HLA, so it was, or transplant or anything. So I was on a steep learning curve, and I had a lot of mentors there that were extraordinary to really help me along my career path.
I think it's very natural for a lot of people over the course of their career or even their studies in one format or another to experience kind of some self-doubt. What's your advice or any sort of tricks or tips that you've discovered as like a workaround for a situation like that when you're sitting and you're just kind of like, I don't know if I can do this?
Well, in that situation, when I went to the blood center, it really did happen. Shortly after I started work, within, I think, the first week or two, the blood center sent me to an international workshop, which is a major meeting in the HLA field where the most recent discoveries of the field are presented, and the experts in the field are giving lectures, and the latest technology is being presented. And I went to that meeting, and I had no idea what anyone was talking about. There was so much jargon. I mean, I was just clueless. So I was blown away. I went back to the blood center, and it was the same thing. I would go to, seminars from the experts in the field at the time, and I wouldn't know what they were talking about. I had my little molecular biology corner, and I was isolating DNA and doing PCR back before there were thermocyclers. In about the 6-month point, I thought this was just such a mistake. I'll never understand. And then suddenly it all cleared up. It just happened. It was just, I think my brain was overwhelmed with all the new information, and it took some time to learn the jargon. Since then, it's just been fabulous.
Yeah, well, I mean, there's a lot that can be said for just recognizing even that things take time. Like most things in life are an adjustment and come with kind of a learning curve. And that's, not something that's just exclusive to careers or anything like that. We like to get into the nitty-gritty, fun, maybe embarrassing things here on Absolute Genius. You don't have to share with us, but we'd love to hear. Over the course of time, what is your most embarrassing lab moment?
I don't have any really dramatic event where something went terribly wrong. No, just, the usual things, because we're all humans and occasionally we pipette something incorrectly. We have, we've pipetted the wrong sample, we've omitted one of the components or something. It happens to everybody. My approach to having that happen is that there's something wrong with the system that allowed me to do it.
Ah, okay.
And so then I focus on what the system is and how did that contribute to my making the errors. So, for my work and most of the people who work with me, we have checklists to prevent those errors. And we take a lot of steps to prevent a person from being able to make a mistake.
Yeah, especially since what you're working with is such a, I mean, it's delicate. It's a rare commodity. It's a, these are these are samples that you really can't mess up with. How about a moment in time when you thought to yourself, wow, I've really made it, or I feel very happy. I feel very content with where I've reached, or I'm super proud.
As time went on and my research continued, I was invited to give lectures around the world to large audiences. I thoroughly enjoy doing that. And some of those crowds were really quite large. And actually looking out on the audience and realizing that they're all totally engaged and excited about what I had done, I would say that would be it.
Amazing.
What an evolution, right? Because we just went over how you went from not having any idea on what a lot of people were speaking about back when you were at the blood center to confident and comfortable to speak to large audiences about all kinds of, really important topics. Like, good for you. What an evolution, right?
It was quite an evolution. Again, when I started out as a graduate student, I thought I would just have a career in academia and do some research and publish some papers. And I really didn't think much about public speaking, but I ended up doing that, and it was a wonderful part of my career.
Yeah, that's excellent. You are clearly balancing a lot. Everybody needs a break from work and research and all these fun things. What do you like to do in your spare time to give yourself a little bit of a mental break from all of this like heavy duty thinking that you're inevitably doing?
I actually don't take many breaks.
You're like, I don't have any time.
This is my idea of fun. I might pick up an issue of science
Okay.
and just read something outside my
Okay.
field. I guess, my husband and I do like gourmet food. So we like going out to restaurants and cooking fun things. But, and then there's family, of course. So that's also a big commitment. I think by the time, you finish with the time for your family and the basics of living and doing research, there isn't any time left, at least for me.
Fair enough. Jordan, do you have any more fun questions? Otherwise, I'm good.
I have two, I not necessarily fun, but two that I was thinking about just talking. Man, we talk about HLA quite a bit in the podcast. Can you describe, just for the listeners and to capture it, what is HLA and how does that play a role in your research?
So HLA is human leukocyte antigen. This is a group of proteins that are expressed on the surface of the cell. They are involved in discriminating self from non-self in the immune system. When you get infected with a pathogen, for example, the HLA proteins bind peptides derived from those pathogens, and this complex of the HLA molecule with the peptide derived from the pathogen is recognized by T lymphocytes, and that's what allows the immune response to target that pathogen and destroy those cells, and ultimately for you to have the infection resolve. The problem with it in transplantation is that now when you transplant the organ, if the HLA proteins of the donor organ or the donor marrow or the donor blood are different, your immune system is going to recognize that as foreign and something to kill. So that's why the HLA matching between the donor and the recipient is one of the important factors in compatibility. That said, now physicians have developed a lot of protocols and their therapeutic alternatives to try to overcome that. So a patient who gets a solid organ transplant will be on lifelong immunosuppression to prevent the immune system from recognizing those differences. I have spent my career, much of it, developing molecular HLA typing methods, chimerism tests to be able to monitor engraftment after blood and marrow transplant. And so my family's well aware of this, and we've been talking about transplant my whole life. And this year, my husband developed a hematological malignancy. And as a result, a lot of the things that I've developed are actually being used for my own family. Really kind of surreal.
Well, it's a good thing you developed them.
Well, I wasn't alone. Those advances are, the product of many people making some little incremental difference, and then it all comes together. But I did play a role in it, and my children grew up with, doing the first sequencing of HLA genes. I used to have them read the autoradiograms. This is when we did P32 labeling. So they helped me read the sequences.
Who needs ABCs when you can do that?
And we were just talking about this a few nights ago, and they said, it was really surreal that they were now having their samples sent for HLA typing, and they could remember how we were working out the technology to get it to work from buccal swabs. And I did a lot of work with the National Marrow Donor Program in developing the registry and getting the donors typed. And my husband's found a matched donor through the National Marrow Donor Program. So, things that I was very involved in are actually influencing us.
It's absolutely incredible, and thank you so much for sharing. On behalf of Cassie and I, Matt and Sarah as well, who are in the background, our best wishes to you and your family. And but what a story, and something that you saw that and thought that you could help improve. Being affected by that now is it sounds surreal, and yeah, but we're with you and your family, and really grateful for all of the work that you've done as well.
Just an excellent discussion. We really, really appreciate your time. This has been great.
Thank you very much for being on Absolute Genius and the podcast. Absolutely enjoyed our conversation. And thank you very much for your time and your personal stories as well. We really appreciate that. We know our listeners do too.
Yeah, and if you can guide people to our digital PCR paper.
You bet.
I am pretty excited about it, and I think it has such great potential, but it's just like, what I talked about in HLA typing. Everybody does some little part, and then it blossoms. And it really takes a village and a lot of people to, make that happen. We've shown it can be done, but there's so much more work to be done.
That was Dr. Leanne Baxter Lowe, Director of the HLA Laboratory at Children's Hospital, Los Angeles. Absolute Genius is produced by Sarah Briganti, Matt Ferris, and Matthew Stock. Thank you so much for joining us for today's episode. We hope you're enjoying Season 2 of our show so far, and that you're as excited as we are for the remaining episodes. Stay curious, and we'll see you next time.
I might have to take a piece of your advice there. I have a 6-month-old. My wife and I just had a baby this year, so he might be getting read some scientific journal articles with
That's
as
right.
well.
Who needs C-spot run when you can just go straight to the scientific journal articles? Forget it. We're just going to skip over it. I love that.
Oh, then you could say, and then look what that virus did. That was a bad virus.
Look what virus did. Yes, I know.
That is amazing.